architecture of the genome versus the transcriptome, maybe run the faster analysis.
Yeah, and it is, as Eric pointed out. I mean, if you have, you know, if a lot of the genome is, and if a lot of your RNA reads include some of those repeats, and some of those repeats may just be trying to map to 10% of the genome. So it'll spend a lot of time doing, you know, mapping to all those sites and then comparing them and trying to decide which is the real site.
Yeah. And so in this case, you're saying repeats as in just like small repeats, not like duplicated sections of the genome?
Yeah, probably.
So like
the parts that are going to be masked out are generally the small repeats.
Gotcha.
And so that's, you know, how you run TopHat. Let's see.
Sorry, if I can just ask.
Yeah.
Something. So I would take my own information and I would put it in there. Did we specify, or does it just automatically output to my folder? Or
Yes.
Okay.
Anything that
I'm sure you already said that, but.
yeah, I don't know. I have the disadvantage that because I was lazy and it's more comfortable for me with my screwed up leg to stay at home a lot of the time. I didn't hear what happened in some of the earlier workshops, but my guess is that they talked about that, but it's easy for it all to, you know, it's a lot of new information at once. But it's important to keep in mind that your data is going to be in your data store section, just as mine is under my username. And you can set up all kinds of folders and whatever structure you want. But your results will always come out in the analyses section. And I can show you that. This is probably a review, but again, it's important. The apps, you know, we started with are found here. Your data is here, and I mean it's pretty obvious. The apps data. The analyses section is very helpful because that's where all your results are. But when you want to find the individual files in your part of the data store, it's under analyses. It's in an independently, you know, created folder for you that says analyses, and each in your analysis section, see, I have TopHat 2 running right now. And if for results of other things that I've run, let's take a look at other TopHat runs. Here's one that I did with mouse. Let's see how that one did. The unfortunate thing with looking at my files is it takes a little bit to pull them up. But you can see this is all within my section of the data store. And you get, this is not necessarily the way command line TopHat will output things. But you'll have a BAM file and an out file, and I think your more interesting information will be in the out file. No, I guess not. I was wrong about that. But these are the raw. This is the raw output if you ran just one file. This output corresponds to that particular read file. And you have these various files, including, you can see, junctions mapping, insertions, deletions, unmapped reads, and then you have your hits, and you have a summary information. Now this is not, again, related to what we're doing today, but this is an example. You have these many input reads, these many maps. Look at that, only 3.3% of the input reads map. I was probably screwing around and mapping it to human or something, I don't know. But if you got that result, that would tell you you're doing something wrong, or you have to know that you are sort of reaching across the evolutionary scale or something. It's 3.3% overall alignment rate is a red flag. But you also have here conveniently named to match your input files, this BAM file, and the index file for that BAM file. And I forgot, the BAM indexes are binary. BAM files are binary. But don't be afraid to look at it, just so you know. It's binary. That's what it's supposed to look like. You don't know anything else. But I look at it sometimes anyway. Just there's stuff in there. It's a binary file. At least I know that much. Because I like to look at my files all the time. I don't like to depend just and on faith that stuff is going on in there. But we'll go back to the community data section, iPlant training. And intro to RNA Seq. And the Top Hat directory gives you the outputs. And so all that information is here, just as it would come out of running Top Hat. But you see that it produces, in this case, just one BAM file, and that's where the formatted outputs are that you will probably want to use for the next step. And let's do that while we're here. So let's snap that to the right. Let's get the analyses window out of the way. It's a little harder on a laptop with a small screen, but it's really handy having that ability to arrange the windows. And so the next thing that we'll want to do, let's see, is the cufflinks. That's the assembler. It links up everything. So Seq. So let's see. Well, the information is in here. Oh, okay. And so all the these are directories. Sorry. Oh, I'm sorry. I keep forgetting. It's the useful stuff is in the BAM directory, or it's supposed to be.
Are you looking for the files that are in that?
Yeah.
To the right, there's, if you just see viewing and like the gray boxes, well, it's my right, I don't know if it's your right.
Oh.
You just,
Oh, okay. I'm not used to using this kind of stuff. I'm used to working on a really big screen. This keeps hiding things. It's difficult. Right. There is where it's hiding. That's why it didn't pop up. Thank you. And this is where you just drag and drop. Just the BAM files. If this is an optional thing here, you can select a reference genome for fragment bias correction. And from my understanding, fragment bias correction is determining which, essentially helping it decide where things actually map on the genome. It will probably, it might take some time to remap some of the touchier areas where there are a lot of options for where related reads could map. But I'm going to skip that. But we definitely need, we should include a reference annotation for comparison. I think that's a pretty good idea. But here again, you don't have to use that from my understanding. And the purpose for that is, again, you're comparing the reference annotation to your information. If you want to push it towards using more your information and less the annotation, then you may decide, no, I don't want to put any of this stuff in there. I want it to work harder at finding new transcripts. I'm just going to leave these as the default. Leave it all default. And launch it. Yes?
I have another question. I'll stop asking you in a little bit, I promise.
No, that's okay.
So
Questions are good.
okay, that won't ever stop. So when you were putting in the annotation information, it was just in a drop-down screen. But if we're going to upload our own file,
Yeah, that was the box right underneath it.
yeah, what file format should that annotation be in? Because I know that people will distribute those in
I think it
tar
can,
balls and all these things.
ideally it's the GTF.
GTF.
I think it may work with GFF. Some of these applications get really, they may start throwing errors if you use a GFF file or certain earlier formats of GFF. It really prefers a GTF, but
And there are conversion software that are available for all that kind of stuff.
well, it's again, depends on what annotation information is available for your genome, or what is produced by the software that you may use to produce the annotation if you're doing it yourself. I think that Maker, for example, which is a program that can do this, can produce a GTF file, but I'm not sure. It can at least produce a GFF file for you. So I think, what time is it? It's
like 12:25.
okay, so, and this ends at 1, so I don't know if I'll have time to go through and show you clicking all these things, but I can do it quickly. But the main thing for you guys to understand is that you can, on your own, go through this, and I recommend going through it on your own, because we don't have time really, as I said before, to go through this and do it, you know, and wait for the results, and then have you look at your results. Ideally, you should do it and see it work and get used to handling the discovery environment. This is a great way of doing it. And if we use Google, we can use the iPlant Learning Center. And you'll see all this really nifty stuff in here. And I think within this are the tutorials. And you'll see a lot of tutorials, and these are really well laid out tutorials to follow. There's one for the tuxedo method. I recommend going through that to learn this method in more detail than I can do with you today. But let me take a little time now to show you what your end results are going to look like, because that's important if you haven't looked at this sort of thing before. The next application, again, this is just dragging and dropping, would have been cuff merge, and that's where you just move all your GTF files that came out of cuff links, move those into cuff merge, merge it with your annotation GTF file, which again, with Arabidopsis, you can do with just a drop down. And you would get your assembly GTF file.
Can you tie it up real quick?
Yeah, I know.
Thanks.
Yeah, that's what I'm doing. And so the cuff diff output would look like this. You get a lot of different files, but among them you can see differential expression. And some of these, it's a column separated format. Yes, no, no meaning no, it's not significant. Yes, it is statistically significant, depending on how you define statistically significant. So these are the results that you'd be looking at. And these are all gene names in this case. Again, these are gene names that you should recognize if you work with Arabidopsis. Pulled right out of the annotation information. You have the same thing for your isoforms.
So in that analysis that has all the isoforms, they averaged out or included, or it just spits out isoforms on the side into a different file format?
Well, I think it includes all the isoforms. I mean, your isoforms, annotated isoforms, should all be in there.
Okay.
It's probably mostly going to be annotated ones with this kind of workflow. And you also have a section where the data comes out, and it's been pre-sorted for you, so you can, and that's pretty handy. But I recommend going through, testing this out for yourself. You should be able to do drag and drop, click and point. And and if you have any questions, I'm available, and I'll turn it over to Erica. I think we might want to take a little break. I'm going to take a break.
Take a couple minutes, but Eric's got to leave by 1, so, or you have to be somewhere at 1.
I need to be upstairs by 1.
Okay, so take just a couple minutes, please, and then we'll